In this tutorial we'll look at protein structures. Proteins are of course built on subunits called amino acids, of which there are around about 20 essential amino acids. Each amino acid has the same basic formula, and what makes them different is a group called the R group, or the functional group, located off the middle carbon. Let's have a quick look. We can see in this diagram here 4 of the different amino acids: methionine, aspartic acid, leucine, and tyrosine. Every amino acid has an amine group, NH2 or NH3 at the terminal end, and a carboxyl group, COO or COOH, at the other end, with a CH in the middle. And then coming off that middle carbon is the functional, or R group. And it's this group that makes each of the 20 amino acids different from each other. A protein is built by bonding amino acids end to end. The bonds that are formed are called peptide bonds. Peptide bonds are formed by the removal of a water molecule, H2O, so that the peptide bond forms between the carboxyl group of one amino acid and the amine group of the next amino acid. As the protein is being built on the ribosome using the triplet code on the messenger RNA molecules, amino acids are put into a particular order based on the messenger RNA code. Each amino acid is brought up to the ribosome by a tRNA molecule, transfer RNA molecule, and peptide bonds are formed. This initial chain of amino acids to form this polypeptide is referred to as the primary structure of the protein. This primary structure in this particular form is still non-functional. The protein cannot do what it's designed to do. The primary structure, as illustrated here, then folds itself into a number of secondary structures which we have studied already. So it forms itself into alpha helixes, represented here, and beta pleated sheets, and sometimes a third structure called random coils. This long chain of amino acids that are then folded into these various secondary structures, depending on which part of the chain we're looking at, then folds itself into what's called its tertiary structure here. For most proteins, this is their final functional shape. This is the shape in which they become active and do their job, whether it's a structural protein or a regulatory protein or a catalytic protein as an enzyme, or whether it's a receptor protein. Whatever the function of the protein is, it generally becomes functional once it's in its tertiary structure. In some instances, a protein may involve the bonding of 4 separate primary chains that have each been folded into a tertiary shape. In this instance, what is formed is the quaternary structure of a protein. So some proteins have a quaternary structure. One classic example is the protein hemoglobin, which is the oxygen carrying molecule that's in the middle of red blood cells. Here's another couple of diagrams showing the 4 protein structures. So we have the primary structure, the long chain of amino acids that are folded then in different sections into alpha helixes, beta pleated sheets, sometimes random coils, that are then folded into the tertiary structure of the protein, which for many proteins is the final functional shape. And in some cases, those tertiary structures, 4 different strands, can be bonded together to form a quaternary structure in some proteins.